Good morning, dear students. Myself, Dr. Arvind Singh Panwar. Today, I will continue the topic pharmacodynamic. Uh, in today's lecture, Pharmacodynamic 2, we are going to study about the receptors and their families. Before that, I would like to discuss about the structural and signal transducer mechanism. It is the mechanism of translation of receptor activation, that is drug action, into functional response, that is drug effect. In other words, it is a complex multi-step process of amplification and integration of signals. This can be subdivided into four major categories that are mentioned below. So here we are going to discuss about the receptor families. So according to type of the molecular structure and the nature of the transducer mechanism, there are four receptor which are also called as superfamilies, which share some common architectures. So these four superfamilies are type 1 channel linked receptors, type 2 that is called G protein coupled receptors, type 3 that is called kinase linked receptor, and type 4 that is called receptors which regulate gene transcription. Now, first of all, out of these superfamilies, all four superfamilies, first three are transmembrane receptors, while the fourth one, that is the receptor which regulate gene transcription, is intracellular in nature. Now, here we are going to discuss about type 1 channel linked receptor. Now, regarding their structure, they are also called ionotropic receptors. They are membrane receptors which are directly coupled, or you can say incorporated with selective ion channels like sodium, potassium, calcium channel or chloride channel. These channels are oligomeric proteins, contain approximately 20 transmembrane segments which are arranged in a circle around central aqueous channel. As you see in the diagram, here you find everything. The functions of this channel, they are primarily involved in the fast neurotransmission only. The complete procedure, for example, ligand bind and opening of the channel takes only milliseconds. The subsequent flow of ion through this channel can elicit a cellular response in the form of hyperpolarization or depolarization of the cell membrane. You see here in the diagram, the N1, N is the agonistic binding site, while COH is the, you can say, domain, channel domain. Now, example of this type of receptors are nicotinic acetylcholine receptor, then 5-HT3, which is also called serotonergic receptor, then GABAergic receptor, and glutamate receptor. So these are the type of receptors. Then type 2 receptor, type 2 G protein coupled receptors. Regarding its structure, you can say they are also known as metabotropic receptor or 7 transmembrane spanning receptor, because all comprise of 7 membrane spanning segment, as you see in the diagram. They are the membrane receptor. In this type of receptor family, one of the intracellular loop is longer than the other and is linked, or you can say coupled to the intracellular effector system through one or more G protein, that is GTP activated protein, for the production of response. The molecules has 7 cylinders, as you see in the diagram, that is membrane spanning alpha helical segment of hydrophobic amino acid. The intervening segment connect these 7 cylinders to form 3 loop on each side of the membrane. So 3 extracellular and 3 intracellular loop. You can see it clearly in the diagram. The amine Means NH2 terminus probably represent the agonistic binding site and is located on the extracellular phase, while carboxy, that is COH terminus, represent the binding site for G coupled protein and is located on the cytosolic site intracellularly. The G protein are floating in the membrane and is composed of three subunits, that is alpha, beta and lambda. The alpha subunit possessing GTPase activity, as you see in the diagram, three components are there. The activation of the receptor results into displacement of GDP by GTP. The active alpha subunit carrying GTP dissociate from other two subunit beta and lambda and therefore activate or inhibit the effector system. Activation of the effector is terminated when the bound GTP molecule is hydrolyzed, which allows the alpha subunit to recombine with other two subunit, that is beta and lambda. So there are several type of G proteins which control different effectors by interacting with different receptor. Few important one are, as you see in the diagram, number 1 GS specific receptor. It stimulates adenylate cyclase, cyclic AMP, which ultimately leads to open calcium channels. The second one is GI specific receptor, G protein coupled receptor, that inhibits, or you can say decrease, adenylate cyclase, cyclic AMP. So alpha 2 receptors have GI receptors and that inhibits postsynaptic adrenergic neuron. Then GQ, that stimulate phospholipase C, that is a part of IP3 DEX system, present in the alpha 1 receptor, have GQ receptor, so calcium in the smooth muscles are released. G0, that inhibits calcium channels, and GI, that opens potassium channel. So these are few specific G receptors proteins. Then target for these G proteins are, number 1 the adenylate cyclase or cyclic AMP system, second one is phospholipase C or inositol phosphate IP3 DEX system, and the third one is the regulation of the ion channels. So let us discuss about the first, that is the adenylate cyclase cyclic AMP system. As you see in the diagram, activation or stimulation of adenyl cyclase mediated by GS protein result in formation of intracellular messenger, that is cyclic AMP, as you see in the diagram. Cyclic AMP activates various protein kinases which control cell function in different way by causing phosphorylation of various enzymes. For example, in heart muscles, cyclic AMP activates protein kinase pKa, which directly or indirectly increase calcium entry into the cells, thereby increasing the force of contraction of the heart muscle. Example, beta receptor in the heart muscle, as you see that on binding this agonist adrenaline to the receptor ultimately leads to stimulate GS, which stimulate adenyl cyclase, and ATP will break down into cyclic AMP, which ultimately stimulate protein kinase. So protein kinase activated is formed, which ultimately causes increase in the glycogen breakdown through glucose. Glycogen inhibit glycogen synthesis and enhances lipolysis, and it also enhances intracellular calcium, so ultimately leads to enhancement in the troponin activity and phospholamban activity. The enhanced troponin ultimately leads to increased contractility of the heart muscle, and increase in the phospholamban leads to faster sequestration or relaxation, which ultimately overall causes increase in the force of contraction of the heart. Then the second one is the phospholipase C, inositol phosphate IP3 DEX system. Now stimulation of phospholipase C, ultimately mediated by GQ protein, result in formation of intracellular messenger like IP3 and that is inositol 1,4,5 triphosphate and GAD, that is DAG, that is called diacyl glycerol from the membrane, phospholipid PIP2, phospholipid phosphatidyl inositol 4,5 biphosphate. Now, this IP3 will act to increase the free cytosolic calcium by releasing calcium from intracellular compartment like tubule. This increases free calcium initiate many actions which includes contraction, secretion, hyperpolarization of the membrane and enzyme activity, while DAC act by activating protein kinase PKC, which ultimately control many cellular function by phosphorylating various proteins. As you see in the diagram here, when this agonist bind with the receptor, that GQ protein gets stimulated, ultimately activate phospholipase C, which ultimately break PIP2 into IP3 and DAC. This IP3 ultimately causes release of the calcium from the storage, while DAG ultimately leads to stimulate PKC. This PKC have number of activities that is incorporated by phosphorylation. While this enhanced or increased concentration of the calcium ultimately leads to binding with the calmodulin, a protein. So calcium calmodulin complex is formed. This calcium calmodulin complex causes activation of myosin light chain kinase. So activated myosin light chain kinase are formed. This activated myosin light chain kinase ultimately causes binding of myosin light chain with myosin and leads to contraction. So in this way, an agonist ultimately contribute in the elicitation of particular response. Then the third one is the regulation of ion channels. So G protein coupled receptor can control the functioning of the ion channels, example calcium and potassium channel, by mechanism that do not involve any second messenger such as cyclic AMP or inositol phosphate, that is IP3. Instead, the G protein directly or indirectly with the channel, thus affecting membrane excitability, transmitter release, contractility, etc. Cardiac muscles, muscarinic acetylcholine receptors are known to enhance potassium permeability. Similar mechanisms is also operates in neuron. So this is about the three types of the receptor proteins. Then type 3 kinase linked receptor. This type 3 kinase linked receptor includes receptor for various hormones, for an example insulin and growth factor. These receptors are totally different as compared to receptor discussed previously in terms of structure and functions. So, structurally, all these receptors contain large extracellular binding domain connected to an intracellular domain via a alpha helix, as you see in the diagram, right? Mechanism. As a result of the agonistic binding to the extracellular domain of these receptors leads to conformational change that result in the dimerization and activation of the intracellular enzyme domain. For an example, tyrosine kinase domain. This results into autophosphorylation, so TKP of the receptors, thus stimulation of that receptors tyrosine kinase activity toward intracellular signaling protein. Here you see in the diagram. Then the last one is the type 4 nuclear receptor or receptor which regulate gene transcription. Here in the diagram it was shown that the receptor mediated regulation of gene transcription is the characteristic property of steroid, thyroid hormone, vitamin D, retinoic acid, etc. These receptors are initially inactive and consist of a conserved DNA binding domain which is attached to variable ligand binding and transcriptional control domain. On binding with a specific ligand drug, they forms agonistic receptor complex which is active. Now this agonist receptor complex moves inside the nucleus as you see in the diagram where the DNA binding domain recognize specific base sequences thus promoting or repressing particular gene, that is gene transcription, and thereby direct the synthesis of specific protein to regulate the activity of the target cells. The effect is slow in onset because they are produced as a result of altered protein synthesis. Here in the diagram you find the complete sequence. Now, signal transduction. So heat stroke proteins normally bind to the nuclear receptor to hold it inactive. The hormone, that is cortisol, sex steroid, tyrosine, bind to the nuclear receptor, releasing the heat shock protein. The hormone receptor complex then bind to the DNA to effector transcription. The cortisol stimulates lipocottin, which inhibits phospholipase A2, which inhibits synthesis of prostaglandin and ultimately play anti-inflammatory properties role. So this is about the receptors. Now we are going to discuss about the efficacy and the potency. There are comparative terms. A drug may be more efficacious or less efficacious than the other, or it may be more potent or less potent than the other. The term efficacy is generally used to compare the therapeutic effectiveness of two drugs. Peak effective or maximum effect that can be produced with the drug is compared. A more efficacious drug will have higher ceiling effect. The term potency is used to compare two drugs on weight-to-weight basis. For an example, if 25 milligram of drug A produces a certain therapeutic response and the drug B can produce the same response with 5 milligram, then the drug B is 5 times more potent than that of drug A. DRC of a less potent drug will be on the right side of the more potent drug. Potency is of little significance in therapeutics because one can give 25 milligram in place of 5 milligram if the same response is obtained. Generally, when the therapeutic effect appears at a higher dose, for example 25 milligram, quite likely the dose dependent adverse effect will also appear at higher dose. A drug producing beneficial action at lower therapeutic dose quite likely will produce adverse effect at that particular lower dose. So, efficacy reflects capacity to generate maximum therapeutic effect, therefore is of more important clinically. Therapeutic window phenomenon. Certain drugs produces optimal therapeutic effect only over a narrow range of plasma drug concentration. Both above and below this range, the beneficial effect of the drug is suboptimal. This is called therapeutic window phenomenon. Example, imipramine, clonidine, glipizide, etc. Similarly, one more term is also important. The term is therapeutic index and it is the ratio of LD50 upon ED50, where this LD50 is the median lethal dose while the ED50 is the median effective dose. This LD50 is the, or you can say this median lethal dose is the dose at which 50% of the population of the experimental animals dies at that dose, while therapeutic ED50 median effective dose is the dose at which the desired response is produced in 50% of the population. Both in LD50 and ED50, percent of population responding is considered. This is not the same as the percent of the maximal response. Therapeutic index indicates safety of a drug. Higher ratio means higher difference between the dose and the lethality and the dose that produces desired effect. Since calculation of the therapeutic index uses lethal dose LD50 and effective dose for 50% of the population, its direct clinical utility is nil. However, it does indicate the safety margin. Clinically more relevant are the MEC, minimum effective plasma concentration, and MSC, that is maximum safe tolerable plasma concentration. For an example, amoxicillin MEC is 5 microgram per ml, while MSC is 20 microgram per ml. In case of phenytoin, they are 10 and 20 microgram per ml respectively. So this completes the first phase of the pharmacodynamics. In the second, or you can say in the third lecture, we are going to discuss about the factors modifying doses and drug action. So thank you very much.